They breathe so rarely that a full minute can pass between one breath and the next. Their body temperature may sit only a degree or two above the air temperature around them. They are, by almost any measure you care to apply, at the very edge of what we mean when we say a creature is alive. And yet in spring, they will fly again. They will navigate. They will hunt insects in darkness, using sonar so refined that it can detect a moth's wing at 30 feet. They will mate and raise young and eat until they are fat again, and then return to this cave, to this chamber, perhaps to this exact position, and do it all again. This is hibernation, but that word, so familiar, so often used casually, to mean nothing more than sleeping through winter, carries within it one of the most extraordinary biological phenomena that life on this planet has ever produced, because hibernation is not sleep, not in any sense. That sleep, as we know it, would recognize. Sleep is a temporary, reversible, easily interrupted change in brain state. You sleep, and your cells continue their ordinary work. Your heart beats 60 or 70 times per minute. Your lungs expand and contract a dozen times. Your body maintains its temperature at 37 degrees Celsius with remarkable precision, burning through energy reserves to keep you warm throughout the night. You wake in the morning, and your body is essentially the same as it was when you lay down. Perhaps a little hungry, perhaps a little stiff, but chemically and physiologically continuous with yourself from the evening before. Hibernation is something entirely different. Hibernation is a controlled biological transformation, a deliberate, genetically orchestrated descent into a state so profoundly altered that some organ systems essentially pause. Metabolism, the engine of life, the sum of every chemical reaction that keeps a cell alive and functional, drops to between 1 and 5% of its normal rate. Body temperature in true hibernators may fall to within a degree of freezing. Breathing slows to once every few minutes or once every few hours in some species. The heart, that most tireless of muscles, beats so rarely that in a ground squirrel mid-hibernation, you might wait 30 seconds between one beat and the next. No surgeon could put a human patient in this state intentionally and expect them to survive. And yet for ground squirrels and bats and hedgehogs and dozens of other species, this state is not an emergency. It is a strategy. It is the solution their lineages arrived at over millions of years of evolutionary pressure to a problem as ancient as winter itself. The problem is this: winter takes away food, and food is energy, and energy is life. What do you do when the source of your energy disappears? For 3, 4, 5 months, you could migrate. Many species do. You could cache food and stay active. Many species do that too. Or you could do something far more elegant, far more radical. You could slow yourself down so completely that you barely need energy at all. You could make yourself into something close to nothing and wait in winter's silence. Life does not disappear. It waits. And in the waiting, there is science enough to fill a lifetime of study, chemistry, and physics, and evolutionary biology, all compressed into the curled body of a sleeping squirrel, breathing once per minute. In the frozen dark, let us stay a little longer. In this forest, let the cold settle around us. Let the pale light grow slowly through the trees. Because what happens in these dens and caves and burrows, while the world above goes white and still, that is the story we are here to tell. Hello, beautiful souls, welcome to this quiet space, a place where minds slow down and hearts rest. Before we begin this gentle, sleepy journey, just a small moment of support. If this meditation brings you calm, comfort, or clarity, please subscribe to the channel, gently tap the like button, and leave a comment to let us know you are here. Your support helps us continue creating these peaceful spaces for your mind, for your heart. Now, there is nothing else you need to do, nothing to think about. Let's begin. Close your eyes now and slowly settle into a comfortable position. Take a gentle breath in and slowly breathe out. Let your shoulders relax, let your jaw loosen, and allow your whole body to feel heavy. Before we go deeper into the biology, we need to clear away a misconception, a very common one that even careful people repeat without thinking. Bears hibernate. You have probably heard this. Bears go to sleep in autumn and wake up in spring. And this is called hibernation. This is true. But it is also, in a very specific scientific sense, not quite right. And the difference between what bears do and what a Siberian ground squirrel does is the difference between a long, restful nap and something that would, if applied to a human body, constitute a medical emergency. To understand hibernation properly, we need to understand what it is trying to accomplish: the conservation of energy at the deepest possible level. And to understand that, we need to understand metabolism. Every living cell is a factory. It takes in raw materials, sugar, oxygen, fats, proteins, and it converts them into energy, into new cellular components, into waste products to be expelled. This process never stops while the cell is alive. Even when you are perfectly still, sitting quietly in a chair, doing nothing, Your cells are running millions of chemical reactions per second. Your liver is processing nutrients. Your kidneys are filtering blood. Your brain is humming with electrical activity. Your immune cells are patrolling your tissues. Your heart muscle is contracting and relaxing with metronomic reliability. All of this costs energy. All of this burns through the fuel that food provides. The rate at which your body burns this fuel at rest, the minimum cost of simply existing, is called your basal metabolic rate. For most humans, this is somewhere in the range of 1,500 to 2,000 calories per day, just to keep the machinery running, with no additional work being done. Now imagine you want to survive for 5 months without eating. At your normal metabolic rate, you would need roughly 200,000 calories worth of stored fat, far more than any human body could reasonably carry. The mathematics simply do not work. A hibernating ground squirrel solves this problem by changing the mathematics. It drops its metabolic rate to somewhere between 2 and 5% of its normal value. Suddenly, the energy budget for 5 months becomes manageable. The squirrel enters autumn, weighing perhaps 40% more than its lean summer body weight. All of that extra mass is stored fat, and it emerges in spring, having burned through most of that fat. Slowly, steadily, over months of nearly suspended animation, this suppression of metabolism is not simply a matter of the animal getting cold and slowing down. That would be ectothermy, the passive temperature dependence of cold-blooded animals like reptiles. Hibernating mammals are still warm-blooded at heart. They have the genetic and physiological machinery to generate body heat. But during hibernation, they deliberately turn down the thermostat. There is a particular kind of silence that belongs only to winter, not the absence of sound exactly, but something deeper, a settling, a holding of breath so complete that the world itself seems to have leaned back into stillness, pressed its shoulders against the cold, and exhaled for the last time before a very long rest. Imagine standing at the edge of a forest before dawn on a January morning. The light has not fully arrived yet. There is only that faint, pale luminescence that winter gives to snowfields, a soft, sourceless glow that seems to rise from the ground itself, rather than descend from any sky. The trees stand bare. Their branches reach upward, like the fingers of something ancient. Dark lines traced across the gray above, holding nothing but frost and silence. The snow beneath your feet is old snow, packed and undisturbed. No wind moves through this place. No birds call. The forest has drawn its curtain closed. You might think nothing lives here now. You might look at the frozen surface of the lake ahead. Its ice thick and opaque, the reeds at its edge encased in crystal, and feel the absolute certainty that this is a dead landscape, a world temporarily evacuated, waiting for warmth to return before life resumes. But you would be wrong. Beneath the snow, not far, perhaps only a few feet below where you stand, there is warmth, there is breath, there is a heartbeat, so slow and so faint that the instruments required to detect it must be pressed very close and held very still. There is a small body, curled against itself, surrounded by dried leaves, and the compressed earth of an autumn burrow. A woodchuck, perhaps, a ground squirrel, some small, round-bodied creature that spent its autumn eating, and sleeping, and eating again, until the fat beneath its skin had grown thick enough to carry it through months of cold. It is not dead. It is not fully asleep. It exists in a state that science has studied for decades and still finds remarkable. A state of such profound metabolic reduction that the body barely resembles anything we would normally call alive. And yet alive it is, waiting, enduring, patiently folded into the earth, while the long, cold mathematics of winter work themselves out above. Walk a little farther through this forest. Find the hillside, where the limestone cliff faces south, and where a narrow crack in the rock opens into a cave chamber, perhaps the size of a small room. In the darkness of that chamber, hanging from the ceiling in clusters, like dusty ornaments, are bats. Dozens of them, perhaps hundreds, depending on the species and the location. They arrived here in autumn, following some internal calendar that requires no external consultation. They navigated to this specific cave and not another, this chamber and not one closer to the entrance, and they hung themselves upside down and let their body temperature fall. Their hearts now beat, perhaps 8 times per minute. In summer, those same hearts beat 400 times per minute. They choose, through a cascade of biochemical signals, to let their body temperature fall to levels that would kill most mammals. And then, they sustain themselves at that reduced temperature for weeks or months. The body temperature of an Arctic ground squirrel in deep hibernation can fall to -3 degrees Celsius. Below freezing, the animal's tissues do not freeze. Certain biochemical adaptations prevent ice crystal formation inside cells. But the body is, by any thermal measurement, colder than a typical refrigerator, and the animal is alive. And in spring, it will warm itself back up through a process called arousal, and it will emerge, a little thin, into the cold but brightening world. This is what scientists mean when they say true hibernation, a dramatic, voluntarily regulated drop in body temperature and metabolism, sustained over a long period, with periodic interruptions, called interbout arousals, followed by a full return to normal function. Bears do something related, but different. A black bear's body temperature during winter dormancy might fall by only 4 to 5 degrees from its summer value. Its metabolic rate drops substantially, but not to the profound depths achieved by ground squirrels or bats. A bear can be woken from its winter sleep with some effort, disturbing a truly hibernating bat or ground squirrel during deep torpor, and they may not wake immediately at all. Bears don't urinate or defecate during their winter dormancy, which is remarkable in its own right. And pregnant bears give birth and nurse cubs while still in their dens, all of which requires considerably more physiological activity than true hibernation allows. Scientists sometimes call what bears do carnivore lethargy or winter dormancy to distinguish it from true hibernation, but in common speech, bears hibernate, and for our purposes here, we will visit them in their dens when we discuss the animals that use winter sleep, because what bears do, while less extreme, is still a biological achievement of considerable sophistication. Then there is torpor, which is worth mentioning now, because it will come up again. Torpor is a short-term version of the same metabolic suppression. A hummingbird that cannot maintain its extraordinary metabolic rate through a cold night will enter torpor, dropping its body temperature dramatically, slowing its heart and breathing, essentially doing overnight what a ground squirrel does over months. A torpid hummingbird on a cold morning looks dead. It hangs from its perch, stiff and cold and unresponsive, but given a little warmth and time, it rouses itself, shivers vigorously to warm up, and resumes its astonishing life as if nothing unusual had happened. Torpor is the daily or short-term version. Hibernation is torpor, extended and sustained across an entire season. The physiological mechanisms overlap enormously. Many scientists now think of them as points on a single continuum. Animals using whatever degree of metabolic suppression their situation requires. What unifies all of these states? Torpor, hibernation, winter dormancy, is the fundamental strategy. When energy is scarce, reduce the demand, slow the engine, turn down the heat, let the body exist at the lowest possible cost until conditions improve. It is a strategy of extraordinary elegance, and it raises an immediate question. How do you slow a body down this much without damaging it? Because the biology of a cell at normal temperature, is precisely tuned to work at normal temperature. Drop the temperature by 20 or 30 degrees, and you might expect proteins to misfold, membranes to stiffen, cellular machinery to seize up. The answer to that question is in the biochemistry, and the biochemistry of hibernation is one of the most beautiful and intricate stories in all of biology. But before we go there, we should ask how this ability appeared in the world at all. Because hibernation did not spring into existence fully formed. It was made slowly by the patient hand of evolution. Close your eyes for a moment and go back, not weeks or years, but millions of years. Go back to the world as it was 65 million years ago, immediately after the asteroid impact that ended the reign of the non-avian dinosaurs. The world was dark for months, perhaps years. Dust and debris blocked the sun. Plants died. The food chains that had sustained the dinosaurs for over 100 million years collapsed. The world grew cold and stayed cold for a long time. The mammals that survived were, almost without exception, small generalists, animals capable of eating almost anything: seeds, insects, roots, fungus, carrion, and crucially, animals with some physiological flexibility, the ability to slow down when food disappeared, the ability to conserve energy when the world became temporarily uninhabitable. The capacity for metabolic suppression is ancient, far older than mammals. Single-celled organisms can enter dormant states. Plants go dormant. Tardigrades, those extraordinary microscopic animals, sometimes called water bears, can enter a state of cryptobiosis so extreme that they survive vacuum, radiation, and temperatures approaching absolute zero. The basic toolkit of metabolic suppression is written into the foundations of life itself. But what mammals did with this ancient toolkit was remarkable. They elaborated it. They refined it. They built regulatory systems around it of astonishing sophistication. And they did so multiple times, independently, in response to the same environmental pressure. The seasonal disappearance of food. Biologists call this convergent evolution. When different lineages, facing the same problem, arrive independently at the same solution, that is convergence. The wing of a bat and the wing of a bird are the same solution to the same problem. Sustained aerial locomotion arrived at along entirely different evolutionary paths. Hibernation is similar. It has evolved separately. in at least 4 or 5 distinct mammalian lineages, including monotremes, marsupials, and multiple groups of placental mammals. This tells us something important. The selective pressure to hibernate is very strong, and the underlying biological toolkit to make it happen is available across a wide range of mammalian body plans. Consider what natural selection actually does. It does not plan. It does not foresee. It has no goal. It simply preserves, generation after generation, the individuals whose traits allowed them to survive and reproduce in a population of small ground-dwelling mammals living in a temperate environment 60 million years ago. Let us say, ancestral rodents in a landscape that had pronounced seasons. Winter would have killed indiscriminately animals that could not find enough food. Through the cold months, died animals that could reduce their energy requirements enough to survive on body fat stores, lived, and passed on whatever genetic variants enabled that survival over millions of generations. This pressure accumulated small mutations in genes controlling thermoregulation, genes controlling metabolic enzyme activity, genes controlling hormonal signaling. All of these were selected for, if they pushed even slightly in the direction of better energy conservation during winter, the animals that could cool down a little and slow down a little survived the cold a little better. Their offspring inherited those tendencies, and the next generation faced the same selection pressure, and the next, and the next. What emerged over vast stretches of time was the sophisticated machinery of true hibernation, genes that regulate when and how the body enters torpor, hormonal signals that trigger the cascade of changes, body temperature falling, heart rate dropping, breathing slowing, metabolism suppressing, biochemical adaptations in individual cells that allow them to survive and function at temperatures that would kill cells. Not so adapted. The ice ages pushed this further. During the Pleistocene epoch, the last 2 million years or so, the era of mammoths and giant ground sloths, and repeated cycles of glaciation, the winters became longer and harsher across enormous swaths of the northern hemisphere. The selection pressure for hibernation intensified. The lineages that had already developed good metabolic suppression were well positioned. Those that had not faced the ice on different terms. There is a reason that the best hibernators today tend to be animals of northern latitudes, ground squirrels of the Arctic tundra, European hedgehogs, dormice in the mountain forests of Central Europe, the little brown bat ranging across Canada and the northern United States. These are the lineages that spent millions of years being tested by serious winters. The ones that passed the test did so because their genes built bodies capable of folding themselves into the cold and waiting. What is particularly interesting to evolutionary biologists is not just that hibernation evolved, but that it evolved in the specific ways it did, because hibernation is not simply turning the body off. It requires active management. The body must be actively cooled, or rather, must actively regulate its own cooling. It must actively suppress certain cellular processes while maintaining others. It must actively preserve tissues that might otherwise deteriorate, and it must actively rewarm when the time comes to emerge. All of this requires the coordinated action of dozens of genes, numerous hormones, and signaling molecules, and metabolic pathways that had to be specifically adapted for the hibernating state. This did not happen in one step. It happened incrementally, each small improvement conferring a marginal advantage, each marginal advantage slightly increasing the probability of surviving the next winter. Evolution is patient in a way that staggers the imagination. It has more time than we can genuinely comprehend, and what it has built in hibernating animals, given that time and that pressure, is one of the most extraordinary physiological systems the life of this planet has produced. Let us go inward now. Let us shrink ourselves down to the scale of a single cell in the body of a hibernating ground squirrel, and look at what is happening there. At the surface of the cell, embedded in the lipid membrane that forms its outer boundary, are proteins, receptors, channels, pumps. These proteins are sensitive to many things: temperature, chemical signals from other cells, the concentration of various molecules in the fluid surrounding them when autumn arrives, and certain hormonal signals change. These membrane proteins begin to alter their behavior. The cell begins to shift. The first and most important shift is in metabolism. Every cell runs on a molecule called ATP, adenosine triphosphate, which is the universal currency of biological energy. Your cells make ATP by burning fuel, primarily glucose and fatty acids, through a process that takes place in the mitochondria. This process requires oxygen. The rate at which your mitochondria produce ATP at any given moment is effectively your metabolic rate. In a hibernating animal, the rate of ATP production in virtually every tissue falls dramatically genes that code for enzymes involved in energy metabolism are down-regulated. The instructions for making these enzymes are still there in the DNA, but they are being read less frequently, so fewer enzyme molecules are produced. So the biochemical reactions they catalyze proceed more slowly. The engine does not stop. It merely idles. But what fuels this idling engine? Not glucose. During hibernation, the primary fuel shifts from carbohydrates to fat. This shift happens in preparation for hibernation. The animal spends the weeks and months before entering the den, eating voraciously, building up enormous fat reserves. In a 13-lined ground squirrel, body fat might increase from 10% of body weight in summer to around 35% by early autumn. This fat is stored primarily in white adipose tissue, the familiar body fat that we all carry, and it will be slowly burned throughout the hibernation period to keep the minimal fires of metabolism burning. But there is another type of fat that plays a crucial role, and it is worth stopping to examine it carefully, because it is one of the most remarkable tissues in the mammalian body. Brown adipose tissue. Brown fat is not like the ordinary fat that stores energy for later use. Brown fat is a furnace. Its cells are packed with mitochondria, far more than ordinary cells. And within those mitochondria is a protein called UCP1, for uncoupling protein 1. This protein does something unusual. It short-circuits the normal process by which mitochondria make ATP. Instead of allowing the energy from burning fat to be captured as ATP, UCP1 allows it to leak away as heat. This sounds wasteful, and in some sense, it is. Brown fat burns a lot of fuel to produce relatively little ATP, but the heat it generates is the point. Brown fat is a biological space heater, and the mammals that use it most intensively are those that need to generate heat rapidly. Newborns, who are small and lose heat quickly, and hibernators, who need to rewarm their bodies from near-freezing temperatures at the end of each hibernation bout. When a ground squirrel begins to arouse from deep hibernation, when it transitions from the near-frozen stillness of deep torpor to the active, warm state of normal function, brown fat is the engine that drives this transition. The squirrel shivers, and its brown fat generates tremendous amounts of heat, and within a few hours, a body that was nearly as cold as the soil around it has warmed to 37 degrees Celsius. This warming process costs energy, It is the most metabolically expensive part of hibernation, actually, but it is accomplished with speed and effectiveness that no external heating system could match. Now let us think about the signals that trigger all of this. How does a ground squirrel know when to begin preparing for hibernation? How does it know when to enter its first torpor bout? The answer involves a complex interplay of hormones, environmental cues, and endogenous biological clocks. As the days shorten in late summer and early autumn, changes in light exposure alter the activity of the suprachiasmatic nucleus, a small region of the brain that acts as the master circadian clock, timing everything from sleep cycles to hormonal rhythms. The changing light pattern causes changes in the release of melatonin from the pineal gland. Melatonin is the molecule of darkness. It is released preferentially at night, and as nights grow longer in autumn, melatonin exposure increases. This increasing melatonin exposure, along with falling temperatures and declining food availability, triggers a cascade of hormonal changes. Levels of certain hormones that suppress hibernation, including thyroid hormones and certain stress hormones, begin to fall. Insulin sensitivity changes. The body's appetite regulation shifts to promote hyperphagia, the eating of extraordinary amounts of food for fat storage, and eventually, when conditions are right and the biological clock says the time has come, the transition into the first torpor bout begins. At the cellular level, this transition involves changes in gene expression so extensive that some researchers describe the hibernating ground squirrel as essentially becoming a different physiological organism. Genes involved in fat metabolism become highly active. Genes involved in carbohydrate metabolism become quieter. Genes that protect cells from the damage that low temperatures and low oxygen can cause. Genes encoding antioxidant enzymes, heat shock proteins, proteins that stabilize cell membranes, are upregulated. The cell prepares itself for the cold, like a sailor preparing for a long voyage, securing everything that might otherwise shift or break. One of the most fascinating aspects of this biochemical preparation concerns the cell membranes themselves. Membranes are made of lipids, fat molecules arranged in a bilayer, and their physical properties are temperature dependent. At warm temperatures, membranes are fluid and flexible. Proteins embedded in them can move freely and function normally. At cold temperatures, membranes stiffen and can become dysfunctional. This is why cold kills most cells. Hibernating animals adjust the lipid composition of their cell membranes. As they prepare to enter torpor, they increase the proportion of unsaturated fatty acids in the membrane lipids. Unsaturated fats, the ones with double bonds in their carbon chains, remain more fluid at lower temperatures than saturated fats do. By adjusting this ratio, the hibernating animal effectively lowers the temperature at which its membranes begin to stiffen, keeping them functional even as body temperature drops towards zero. This is biochemistry deployed with the sophistication of an engineer. The animal does not think about it, does not plan it, is not consciously aware of it at all. It is written into the genetic instructions that respond automatically to the hormonal and environmental signals of approaching winter. The cell, in all its microscopic complexity, prepares itself for the cold, and then the cold comes, and the cell waits. There is a question that stops many people when they first think carefully about hibernation, a question that seems to have an obvious, alarming answer, and yet does not. If the body temperature of a hibernating ground squirrel falls to near freezing, if its heart beats 4 or 5 times per minute instead of 300, if the electrical activity in its brain diminishes to a whisper, then why doesn't it die? Our understanding of medicine creates a specific expectation here. We know that if a human heart slows too dramatically, if it is deprived of oxygen for more than a few minutes, if blood pressure falls below certain thresholds, catastrophic and irreversible damage occurs. The brain, the most oxygen-hungry organ in the body, begins to die within 4 to 6 minutes of interrupted blood supply. Cardiac arrest leads to brain death, leads to death. This is the hard limit of human physiology under cold or oxygen-deprived conditions. And yet, the ground squirrel's heart beats 5 times per minute for months, and no damage occurs. The brain sustains itself on a trickle of blood and oxygen. The kidneys and liver slow to near stillness, and then everything reverses, and the animal walks out of the den in spring, apparently none the worse for its extraordinary winter. How? The answer unfolds at multiple levels, from the molecular to the organ system level, and each level reveals another layer of the remarkable adaptation that hibernation represents. Begin with the heart itself. The cardiac muscle cells of hibernators have specific biochemical adaptations. They allow them to function at low temperatures and low metabolic rates. They alter the isoforms, the specific molecular variants of key proteins like myosin, the motor protein responsible for muscle contraction. The low temperature isoforms of these proteins contract and relax efficiently at temperatures that would make the normal isoforms sluggish and dysfunctional. The heart of a hibernating animal is not the same heart, biochemically speaking, as it is in summer. The blood itself changes. The viscosity of blood increases. As temperature falls, it becomes thicker, harder to pump. Hibernating animals compensate by reducing the hematocrit, the proportion of blood, that is red blood cells, as they enter torpor. A thinner, more watery blood is easier to move through narrowed, cold-stiffened vessels. The trade-off is reduced oxygen carrying capacity, but at such low metabolic rates, the demand for oxygen is so low that reduced carrying capacity is not a problem. The blood pressure management during hibernation is particularly extraordinary. In the deep phase of torpor, blood pressure falls dramatically, and yet the organs do not suffer the kind of damage that low blood pressure causes in non-hibernating mammals. This seems to involve a kind of pre-adaptation of the vascular system. The blood vessels of hibernators are specifically tolerant of the low flow conditions of torpor in ways that human vessels are not. Perhaps most remarkable is what happens in the brain. The brain is the organ we most fear losing to cold or hypoxia. Its enormous metabolic demands, its absolute dependence on continuous oxygen and glucose supply, its catastrophic vulnerability to even brief interruption of blood flow. All of this makes the brain seem like the weakest link in any attempt to slow a body down dramatically. And yet, the brains of hibernating animals not only survive deep torpor, but appear to benefit from it in certain ways. During torpor, neural activity, the firing of neurons, the transmission of signals across synapses is dramatically reduced. Many of the brain's normal activities essentially pause. The electroencephalogram of a deeply torpid ground squirrel shows a pattern approaching flatline, a pattern that in a human would indicate profound brain damage or death. But the neurons are not dying. They are being protected by a suite of molecular mechanisms so sophisticated that neurologists and neuroscientists have spent decades trying to understand them. During torpor, neurons upregulate the production of proteins that protect against the stresses of low temperature and low oxygen heat shock proteins that prevent protein misfolding, antioxidant enzymes that neutralize the damaging molecules produced by low-level metabolic activity, proteins that stabilize cell membranes and cytoskeletal structures. There is also a fascinating phenomenon involving synapses, the connections between neurons through which nerve signals pass. In some hibernating species, the actual number of synaptic connections in the brain decreases during deep torpor and then reforms during arousal. The brain, in a sense, partially disassembles its own wiring during the most extreme phase of hibernation and then reassembles it when warming. This is the kind of observation that seems impossible until it is documented repeatedly and thoroughly, and it remains one of the most extraordinary aspects of hibernation neuroscience. The distinction between hibernation and coma is worth making gently here. A coma is a pathological state. Something has gone wrong. Something has disrupted normal function. The brain in a coma is damaged or suppressed by injury, toxins, or disease. Hibernation is the opposite. A precisely regulated, genetically programmed reduction in neural activity that the brain itself orchestrates. The hibernating brain is not being damaged into a lower activity state. It is deliberately choosing one. This distinction has enormous implications for medicine. For decades, physicians and researchers have been fascinated by the possibility of inducing a hibernation-like state in human patients, not as a curiosity, but as a potential tool for saving lives. Consider trauma surgery. The time pressure in major trauma, the frantic race to repair a damaged heart or aorta before the patient bleeds to death or suffers irreversible brain damage is one of the most intense challenges in medicine. If surgeons could rapidly cool a patient and reduce their metabolic demands, buying additional time to perform complex repairs without the clock of oxygen deprivation ticking away, the outcomes could improve dramatically. Some research groups have explored emergency preservation and resuscitation techniques that use rapid cooling to approach this goal, or consider organ transplantation, the window of time during which a donor organ can be preserved outside the body before transplant is severely limited by the damage that accumulates during cold storage. If the cells of that organ could be induced to enter something like torpor, minimizing their metabolic demand, protecting themselves against the stresses of cold and low oxygen, preservation time could potentially be extended significantly, expanding the geographic range over which organs could be transported and matched to recipients. The heart that almost stops is teaching us something about the flexibility of life, the range of conditions under which a body can sustain itself. In our fascination with the extremes of human performance, the fastest runners, the strongest lifters, the highest achievers, we sometimes forget that one of the most extraordinary performances a body can give is the performance of stillness, of radical self-reduction, of enduring through the cold and the dark until the time for activity returns. The ground squirrel's near-stopped heart is not a failure. It is an achievement, and it beats. It's 5 slow beats per minute in the dark, and it is sufficient. Let us travel for a while. Let us visit the places where hibernation happens, and the creatures who have made it their strategy for surviving winter's long claim on the world. Begin in the grasslands and prairies of North America, where the 13-lined ground squirrel, small, striped, alert in summer, absent in winter, digs its burrow in early autumn. This animal is one of the most studied hibernators in science, partly because of its accessibility, and partly because the hibernation it undergoes is among the most dramatic in the mammal world. A 13-lined ground squirrel, entering its first autumn hibernation bout, drops its body temperature to between 1 and 7 degrees Celsius. Its heart rate falls to perhaps 3 to 5 beats per minute. It breathes once every few minutes. It will stay in this state for 2 to 3 weeks, then spontaneously warm itself to near normal body temperature for a day or two, spending enormous energy on this arousal, curiously, before dropping back into torpor. It will cycle through this pattern perhaps 20 or 30 times before spring finally arrives, and it emerges permanently. Why do hibernators arouse periodically? This is one of the enduring mysteries of hibernation biology. The arousals cost energy, enormous energy, relative to the deep torpor state, and they seem to defeat part of the purpose of hibernating. Several hypotheses have been proposed. Some evidence suggests that arousals are necessary for certain immune functions. The immune system operates poorly at very low temperatures, and periodic warming may allow the animal to clear cellular debris and maintain immune surveillance. Other evidence suggests arousals may be needed for memory consolidation or for certain repair processes in DNA and proteins that require normal temperatures to function. The honest scientific answer is that we are still not certain. The periodic arousal remains a puzzle, wrapped inside a winter. Now move to the caves. Europe and North America both host large bat hibernation colonies in limestone caves, and visiting one in winter is to encounter one of the most haunting and beautiful sights in natural history. Dozens or hundreds, or sometimes thousands of bats clustered together in the darkness of underground chambers, their bodies densely packed for warmth. Little brown bat, brown long-eared bat, greater horseshoe bat, species by species, each claiming its preferred zone within the cave, some preferring the deepest and coldest areas, some closer to the entrance, where it is slightly warmer. A hibernating bat, hanging in a cave, has a damp film on its fur. The condensation of the cave air on its cold surface. Its wings are folded tight against its sides. The leathery membrane that in flight is stretched across such elegant elongated finger bones, now wrapped like a self-made blanket. It will hang here for months, and in that time it may lose a third of its body weight. The fat it accumulated in autumn, eating hundreds of insects per night through the warm months, is almost gone by spring. The timing of emergence is critical. Come out too early and there are no insects. Stay too long and the fat runs out. The bat navigates this window by a combination of external cues and internal physiology, by the temperature of the cave air, and by some deep biological clock that keeps a rough count of the season. Bears. We must speak of bears, because they are the animal most associated in most minds with winter sleep, and because what they do, while distinct from true hibernation, is no less impressive in its own way. A pregnant American black bear enters her den in late autumn and does not emerge until spring, a period of perhaps 5 to 7 months. During this time, she does not eat, drink, urinate, or defecate. She gives birth, usually to 2 cubs, in January or February, and she nurses those cubs throughout the remaining months of her den time, providing them with milk rich enough to sustain their rapid growth. All of this is accomplished on fat reserves accumulated in autumn. The metabolic rate of a denning bear drops to about 50 to 60% of her summer rate. Substantial suppression, though nothing like the 95% reduction of a true hibernator. Her body temperature falls by 4 to 6 degrees rather than 30. She can be woken with some effort, but the fact that she recycles her waste products internally during denning, reabsorbing urea and other nitrogenous compounds that her kidneys would normally excrete, converting them into usable amino acids, is a biochemical feat that physiologists still find remarkable. Her muscles remain strong and functional despite months of inactivity, because bears have evolved specific mechanisms to prevent the muscle wasting that would afflict a human in similar circumstances. We will return to that muscle preservation in a later chapter, because it has implications that reach beyond bears and into medicine and aging research. Hedgehogs. In the gardens and hedgerows of Europe, these prickly, endearing animals find themselves a nest of leaves under a fallen log or a garden shed, and they curl themselves into a ball and do something that observers sometimes mistake for death. A hibernating European hedgehog, curled in its nest, is profoundly unresponsive, cold to the touch, barely breathing. Its body temperature can fall to 4 degrees Celsius. Its heart rate drops from about 190 beats per minute in summer to around 20 in hibernation. It breathes very rarely. It will spend perhaps 5 to 7 months in this state, depending on the severity of the winter and the latitude of its location. The hedgehog's hibernation is less perfectly regulated than that of some true hibernators, which makes it both more vulnerable and more interesting to study. On mild winter days, it may partially rouse, occasionally even emerge from its nest to forage briefly before returning. It is sensitive to disturbance in a way that some more committed hibernators are not. These characteristics have made hedgehogs useful models for understanding the transition states between full torpor and wakefulness. And then there are the frogs. Wood frog of North America has one of the most extraordinary winter strategies of any animal. They do not hibernate in any conventional sense. They freeze, not partially, not metaphorically, literally freeze. Ice crystals form in the spaces between their cells, and the frog's body becomes rigid. Its heart stops. Its brain activity ceases. By any ordinary definition, the wood frog in midwinter is dead. And yet, in spring, it thaws. Its heart resumes beating, and it hops to the nearest pond to breed. The mechanism that allows wood frogs to survive freezing is a story of biochemical ingenuity that deserves its own lengthy chapter. For now, it is enough to note that the wood frog represents the outer limit of what we might call suspended animation, a complete cessation of all vital signs, and that this too is a strategy for surviving winter, perhaps the most radical one that vertebrates have evolved. Different animals, different depths of stillness, different physiological mechanisms, but the same ancient problem and the same ancient answer. Winter is long, food is scarce, and the body that can wait will survive. Picture the smallest bird you have ever seen, perhaps a wren darting through underbrush, or a kinglet barely the size of a large thumb. Now imagine something smaller still. The bee hummingbird of Cuba, which weighs less than 2 grams, less than a dime, and is the smallest bird in the world. Hummingbirds, regardless of species, face a particular metabolic challenge that illuminates something fundamental about the relationship between body size and energy use. A hummingbird's metabolism is so fast, so furiously energetic, that it must eat almost constantly while active. The hovering flight that makes them such astonishing creatures. That ability to hang suspended in air, wings beating 50 to 80 times per second, costs more energy per unit body mass than almost any other form of locomotion in the animal kingdom. A hummingbird's heart beats 1,200 times per minute. During active flight, it breathes over 200 times per minute. It processes enormous quantities of nectar and converts them to energy at a rate that strains the imagination. Sustain this through a cold night, when nectar is unavailable and body temperature needs to be maintained, and the hummingbird would burn through its entire energy reserves before morning. It would die of energetic exhaustion, so it does not try. Instead, as darkness comes on a cold night, a hummingbird settles onto a perch and allows its body temperature to fall, not by a degree or two, but dramatically, from its normal body temperature of around 40 degrees Celsius to perhaps 15, or sometimes lower. Its heart rate drops from 1,200 beats per minute to perhaps 50. It breathes so slowly as to be almost undetectable. It enters torpor. In this torpid state, the hummingbird can survive the night on a tiny fraction of the energy reserves that normal thermoregulation would have required. When the morning warms and light returns, it warms itself using the same kind of brown fat thermogenesis that hibernating mammals use, and resumes its extraordinary, frenetic life. This daily torpor is not the same as hibernation, but it is the same phenomenon at a different time scale. And understanding the relationship between torpor and hibernation requires understanding something about body size that has profound implications across biology. Smaller animals lose heat faster than larger animals. This is a consequence of geometry. As an object gets smaller, its surface area decreases more slowly than its volume. A mouse has much more surface area relative to its body mass than an elephant does, and so the mouse loses heat to its environment much more rapidly to maintain a constant internal temperature. The mouse must burn fuel much faster per gram of body weight than the elephant. This scaling relationship explains why the smallest mammals have the fastest metabolisms and why they are most susceptible to the energetic crises that cold or famine create. A shrew, one of the smallest mammals, eats nearly its own body weight in food every day. It cannot afford even a brief interruption of food supply. It cannot afford to sleep long. It must eat constantly to keep its internal furnace burning. Torpor is the metabolic pressure valve for small animals. When the cost of maintaining normal body temperature exceeds the energy available, drop the set point. Let the temperature fall. Reduce the metabolic demand until the crisis passes. The same logic that governs nightly torpor in hummingbirds governs daily torpor in small bats that wake to find a cold, insect-free night. It governs multi-day torpor in small desert rodents facing a week of particularly cold temperatures. It governs the winter hibernation of ground squirrels and dormice. The depth and duration change, but the mechanism is the same. There is a cascade of consequences to this understanding. It tells us why bears, despite being the most celebrated hibernators in popular culture, do not undergo the profound torpor that smaller animals do. A bear's large body mass means it loses heat slowly. It can afford to maintain a temperature only a few degrees below normal without burning through its fat reserves too quickly. A ground squirrel, small and heat-losing, must drop far lower to save enough energy to survive. The smallest true hibernators, certain species of dormice, experience the most extreme torpor, their body temperatures falling within a degree or two of their immediate environment, their metabolic rates reduced by 97% or more. These animals exist at the far end of the metabolic suppression spectrum, and studying them has provided some of the deepest insights into how life can be slowed almost to nothing and still maintain its essential structure. Torpor, then, is not a lesser form of hibernation. It is the same response, calibrated to the same problem, expressed at the scale that each animal's size and ecology require. It is the daily negotiation that small animals make with the cold, the same negotiation that becomes seasonal in hibernators, the same ancient strategy, dressed in different temporal clothing. There are places on this planet where the survival problem is not merely difficult, it is, by any reasonable assessment, impossible. And yet life has found its way there too. And where life has gone, hibernation has often followed, pushed to the edges of what biology can accomplish. The Arctic tundra in January, not the forgiving Arctic of summer documentaries, with its light-filled nights and extravagant wildflower meadows. January Arctic, minus 40 degrees Celsius, in a landscape scraped bare by wind, the ground frozen to depths of hundreds of meters, darkness for 20 hours of every day, and a silence so complete that your own heartbeat sounds loud. Beneath the surface of this landscape, Arctic ground squirrels sleep, not merely in shallow burrows, but sometimes at depths of a meter or more, below the permafrost surface, insulated to some degree by snow and frozen earth, but still exposed to temperatures that would kill most mammals in minutes. Arctic ground squirrel body temperatures during deep torpor have been measured at -3 degrees Celsius, below the freezing point of water, below the freezing point of most biological fluids. The squirrel's tissues do not freeze, but that they do not freeze at -3 degrees is itself a profound puzzle, because the water in cells normally freezes well above that temperature. The solution is a combination of factors: the very high concentration of solutes in the blood and cellular fluids, which depresses the freezing point, specialized proteins that interfere with ice crystal formation, and a suite of other adaptations that are still being studied. These squirrels are not simply cold tolerant. They are operating at a biophysical limit that most life on Earth does not approach. Now go higher to the high-altitude meadows of Central Asia or the Andes, where marmots live in colonies and dig burrows that must serve as winter retreats. At elevations above 3,000 meters, winters are severe and very long, sometimes 8 or 9 months of snow and cold. The marmot's hibernation period is proportionally extended. A yellow-bellied marmot in the Sierra Nevada might hibernate from late summer until May. At higher latitudes or altitudes, the hibernating season can be even longer. What is extraordinary about high-altitude hibernators is not just the duration of their winter sleep, but the fact that they must accomplish their entire annual food intake. The hyperphagia that builds their fat reserves in a compressed summer window that may be only 3 or 4 months long. The time pressure is intense. These animals eat with a kind of focused desperation. Through summer, their biology essentially prioritizing fat storage over everything else, because the fat they accumulate in those few months must sustain them through the 8 or 9 that follow. Now consider the opposite extreme. Heat, estivation, the summer equivalent of hibernation, occurs in animals that face not winter cold, but summer heat and drought. The lungfish of Africa burrows into the mud of a drying lake bed, secretes a mucus cocoon around itself, and can survive there for years if necessary. Its metabolism reduced to a fraction of normal, waiting for rain to fill the lake again and restore the conditions it requires for active life. This is metabolic suspension, in the service not of cold survival, but of desiccation survival. The same fundamental strategy, serving a different environmental extreme. Certain species of land snails in Mediterranean climates seal themselves inside their shells with a dried mucus plug and enter torpor through the dry summer months, reactivating only when autumn rains bring moisture. desert frogs in Australia can remain buried and dormant for multiple years. These are animals pushing the principles of hibernation to their absolute limits. But perhaps the most extraordinary cold weather survival story in the vertebrate world belongs to the wood frog, which we mentioned earlier, but which deserves to be examined more closely here, because what it does is genuinely beyond what most people believe biology is capable of. In autumn, wood frogs seek shelter under leaf litter in forests across the northeastern North American continent. When the temperature drops below freezing, ice begins to form, first in the spaces between the frog's skin and the leaf litter, then penetrating into the body itself. Ice crystals form in the fluid between cells. The frog's blood stops circulating. Its heart stops. Its brain goes dark. By any ordinary physiological measurement, the wood frog is dead. The frog survives this because it has an extraordinary biochemical preparation. As winter approaches and the first freezing temperatures arrive, wood frog cells rapidly accumulate enormous quantities of glucose, ordinary blood sugar, but at concentrations that would be catastrophically diabetic. In any other context, this glucose acts as a cryoprotectant, preventing ice from forming inside the cells themselves. Ice forms outside the cells, in the intercellular spaces, and the cells remain liquid and metabolically suppressed, but not frozen. The structural proteins of the frog cells are also specifically adapted to remain functional in a partially frozen context. Specialized ice-binding proteins help control where and how ice crystals grow, preventing them from forming large, sharp structures that would puncture cell membranes. When spring arrives and temperatures rise, the ice melts, the blood resumes flowing, the heart resumes beating, and the frog hops off to find a mate. The process takes only a few hours. The frog that was, by every measurable criterion, biologically dead for months, is alive again. This is biology operating at the edge of what physics allows. Life is maintaining itself in conditions that, by most theoretical frameworks, should preclude it entirely. And it does so, not through any mystical principle, but through evolution's patient, iterative refinement of molecular machinery, the right proteins, present in the right concentrations, doing the right things at the right times. If life can do this in a small amphibian in a forest in Vermont, the question naturally arises: what else might it be capable of? What else is possible? within the laws of physics and chemistry. For a sufficiently evolved biological system, the extreme hibernators, the frozen frogs, the supercooled squirrels, the lung-suspended lungfish, are not curiosities. At the margins of biology, they are demonstrations of what life can be. No animal wakes up one morning and decides to hibernate. No squirrel looks at the falling leaves and thinks, well, time to head underground. The decision, if we can call it that, is made long before, by systems the animal has no conscious access to, running on a schedule determined by light and time, and the deep programming of evolution. The question of how animals know when to hibernate is, at its heart, a question about biological timekeeping. And biological timekeeping is one of the most elegant phenomena in all of biology. Every living cell contains a molecular clock. This is not a metaphor. It is a literal biochemical oscillator, a set of proteins that interact with each other in a cycle that takes approximately 24 hours to complete. These proteins include molecules with names like clock and BMAL1 and period and cryptochrome, and their interactions form a feedback loop. Some proteins activate the production of others, which then inhibit the first, which causes their own levels to fall, which allows the first proteins to become active again in a cycle that ticks away with remarkable precision in almost every cell of your body. This is the circadian clock, from the Latin for about a day, and it coordinates the timing of virtually everything in mammalian physiology: sleep and waking, hormone release, body temperature fluctuation, immune activity, metabolism. Nearly every cell in your body knows what time of day it is, but circadian clocks, precise as they are, run slightly fast or slow relative to the actual 24-hour day. They need to be reset regularly. The primary resetting signal in most animals is light, specifically light detected by a specialized set of cells in the retina that project directly to the suprachiasmatic nucleus of the hypothalamus, the brain's master clock. Every morning, light resets the clock. This is why jet lag occurs. When your clock and your location's light schedule become temporarily misaligned. For hibernating animals, the relevant signal is not just the time of day, but the time of year. The brain needs to know not just that it is morning, but that it is September morning, rather than June morning. The primary cue for this annual timing is photoperiod, the length of the day, or equivalently, the length of the night. As summer wanes and autumn approaches, the nights grow longer. This change in day length is detected by the retina and transmitted to the pineal gland, which responds by releasing melatonin for progressively longer periods each night. Melatonin is thus both a signal of darkness and a signal of the season. The longer the melatonin pulse, the longer the night, the later in the year it must be. This melatonin signal, acting on specific receptors throughout the brain and body, triggers the cascade of preparation that precedes hibernation. Appetite changes. The animal enters the hyperphagia phase, eating voraciously to build fat stores. Behavior changes. Den building or nest preparation activity increases. Hormone levels change. Reproductive hormones drop, since winter is no time to raise young, while hormones associated with fat storage and metabolic depression increase. The interesting thing is that this process begins weeks or months before the animal actually enters its first hibernation bout. A 13-lined ground squirrel, living in a laboratory under constant temperature and constant food availability, will still begin its annual preparation for hibernation at the appropriate time of year, driven by light cues alone. The biology is that deeply programmed, and the preparation is detailed. The hyperphagia of autumn is not indiscriminate eating. Hibernating animals specifically seek out and preferentially eat foods with the highest fat content, seeds and nuts, and protein-rich foods that will build the specific kinds of fat reserves their bodies need. Some species also cache food in their dens for use during periodic arousals, though fat-fueled true hibernators tend to cache less than non-hibernating species. Den building or nest selection behavior intensifies. Bats return to their traditional hibernation caves, sometimes traveling hundreds of kilometers. Ground squirrels deepen and prepare their winter burrows, lining them with dried grass. For insulation, bears begin their search for den sites in late summer, and a pregnant bear's den selection is particularly thorough. She needs a site that will remain undisturbed and well insulated for the months she will spend nursing cubs who cannot thermoregulate for themselves. The body itself prepares. The composition of body fat changes. The fatty acid profile shifts toward the more unsaturated forms that will remain fluid at low temperatures and be metabolically accessible. During hibernation, levels of certain proteins in the blood change. The immune system is pre-adjusted. The liver stocks up on particular enzymes. All of this is orchestrated by the clock inside. The melatonin signal, reading the seasonal light. The hypothalamus interpreting that signal and coordinating the preparation. And every cell in the body responding to the cascade of hormonal and neural signals that follow. The animal does not think its way into hibernation. The clock inside does not consult the animal. It simply tells time, and the body follows. There is something quietly wonderful about this. The apparatus of preparation is running months before the need arrives. The body is already responding to a winter that has not yet come, building the resources and making the adjustments that will be needed when the cold finally arrives. It is a kind of biological foresight, not conscious, not intended, but effective beyond any conscious planning system that a brain could devise. The clock inside ticks on. The nights grow longer. The fat accumulates under the skin. And somewhere in the back of the squirrel's burrow, the leaves it gathered for bedding wait. Nothing in biology is free. Every adaptation carries costs. Every strategy that solves one problem creates others. And the constraints of physics and chemistry ensure that no solution is perfect. Hibernation is among the most powerful survival strategies evolution has produced, and it comes with a suite of genuine costs and risks that hibernators must navigate carefully. The most obvious cost is muscle loss. When a vertebrate animal is immobile for extended periods, the muscles atrophy. The proteins that make up muscle fibers are continuously broken down and rebuilt as part of normal cellular maintenance. When the signals that drive muscle use, the nerve impulses, the mechanical stress of contraction, are absent for weeks or months, this rebuilding process slows, and the muscles progressively weaken and shrink. In humans, muscle atrophy from prolonged immobility is a significant medical challenge. Patients in intensive care, astronauts in zero gravity, elderly individuals with limited mobility, all face the problem of muscle wasting from disuse. And yet, bears emerge from 5 months of winter denning with their muscles largely intact. Ground squirrels that have been curled motionless in a burrow since October can run and climb and dig in May. How? The answers appear to involve several mechanisms that are still being studied. Hibernating animals appear to maintain higher levels of certain molecules that suppress muscle protein breakdown. They actively counteract the catabolism that would normally consume unused muscle. Some species show evidence of periodic muscular contractions during hibernation, possibly driven by shivering thermogenesis during arousal phases, which may provide enough mechanical stimulus to maintain muscle structure. Bears, whose winter dormancy is less metabolically extreme than true hibernation, may be active enough during their denning period, turning, nursing cubs, occasionally moving, to prevent the worst muscle loss. The biochemistry of this muscle preservation is of enormous interest to medical researchers working on everything From space medicine to aging, finding the molecular switches that allow bears to maintain their muscles through months of inactivity could point toward therapies for patients with muscle-wasting conditions. Bone presents a similar puzzle in non-hibernating animals and in humans. Extended periods of inactivity lead to bone density loss. The skeleton depends on the mechanical stress of weight-bearing and muscle activity to maintain its density and strength. Astronauts in space lose bone density rapidly. Patients confined to bed lose it more slowly. Over months, this loss accumulates significantly. Hibernating bears do not lose meaningful bone density during their winter dens. Again, specific biochemical mechanisms appear to suppress the normal bone resorption process during hibernation, maintaining skeletal integrity. Despite the absence of mechanical loading, the identification and understanding of these mechanisms is an active area of research with direct applications to human osteoporosis treatment. There are other costs, less immediately obvious. The immune system operates suboptimally at low temperatures and reduced metabolic rates. The reduced immune surveillance during deep torpor means that pathogens and parasites that normally would be recognized and eliminated may gain a temporary foothold. Some hibernating animals show evidence of increased infection burden at the point of spring emergence, suggesting that their immune defenses were indeed compromised. Through the winter, this immune vulnerability is tragically illustrated by the devastating disease that has ravaged bat populations across North America. White nose syndrome, caused by a cold-adapted fungus that thrives at the temperatures of hibernation caves, infects the skin of hibernating bats. During winter, when their immune systems cannot mount an effective response, by the time bats emerge in spring, the infection has caused severe tissue damage, disrupting thermoregulation and energy metabolism. The mortality rates in affected colonies have been catastrophic, some populations losing 90% of their members in a single winter. The link between the costs of immune suppression during hibernation and the vulnerability to this specific pathogen is tragically direct. Climate change introduces additional risks to an already carefully balanced system. Hibernating animals have evolved to synchronize their winter sleep with the actual conditions of winter. The timing, temperature, and duration of cold that their environments reliably produce. When those conditions shift, the mismatch between the animal's internal programming and the external world can become dangerous. A bat that emerges from hibernation in February because unusually warm temperatures have disrupted its cave's microclimate may find no insects available It is still too early. If it cannot retreat into hibernation again, its fat reserves will be exhausted before food is available. A ground squirrel that cannot find enough food in a shortened autumn to build adequate fat reserves may not have enough energy to survive until spring. A marmot whose high-altitude snowpack melts earlier than usual may emerge to find that the grasses it depends on have already seeded and dried, reducing its hyperphagia window. These are not hypothetical risks. They are being observed now in populations of hibernating animals across the temperate and subarctic world. As the patterns of winter change faster than evolution can keep pace with, but the risks, real as they are bounded. Millions of years of natural selection have built hibernating animals that are robust and flexible within a certain range of conditions, and some species show remarkable capacity to adjust their hibernation timing and duration in response to changing conditions, suggesting that adaptive flexibility exists and that evolution is already working on the problem, as it always has. The costs of hibernation do not negate its power as a strategy. They simply remind us that even the most elegant biological solution is a compromise, a best available answer to an impossible question, maintained at some cost, functional within a range of conditions, and continually refined by the patient pressure of the world. For as long as humans have observed animals sleeping through winter, we have wondered, could we do this too? Not as a survival strategy. Human physiology does not support natural hibernation, and we live in ways that make winter starvation unlikely in most of the world today, but as a medical and physiological tool, as a way of solving problems that currently lie at the boundaries of what medicine can accomplish. The answer, in increasing measure, appears to be yes. Not yet, and not easily, and not without significant scientific challenges still to overcome. But the direction of the research is clear, and what hibernating animals have demonstrated is that the metabolic suppression we observe in them is not confined to certain uniquely evolved lineages. The underlying mechanisms appear to be available in modified form across a much wider range of mammalian species, potentially including us. Let us begin with what is already known. In the late 2000s, researchers at the University of Washington published a series of papers describing how exposure to hydrogen sulfide, a toxic gas at high concentrations, but active as a signaling molecule at very low concentrations, could induce a reversible state of metabolic suppression in mice. Mice that inhaled small amounts of hydrogen sulfide dropped their oxygen consumption by 90%. Their body temperature fell significantly, and their heart rate slowed dramatically. When removed from the gas, they returned to normal physiology. without apparent harm. This was not quite hibernation, but it demonstrated something profound. That mouse physiology, which does not naturally include hibernation, contains the underlying mechanism to enter a significantly suppressed metabolic state. When given the right molecular trigger, the pathway was there. It just required the right key to unlock it. Other research has identified specific regions of the brain that appear to control the onset of torpor. A small cluster of neurons in the hypothalamus, a region involved in temperature regulation and energy balance, when artificially activated, can drive mice into a state remarkably similar to torpor. These neurons project to other brain regions involved in thermoregulation and metabolism. Their activity drops core temperature, slows the heart, reduces oxygen consumption, and when their activation ceases, the animal returns to normal. The switch for metabolic suppression, it appears, exists in mouse brains. It may exist in human brains as well. This has immediately obvious implications. For medicine, the ability to reliably induce a controlled, reversible state of profound metabolic suppression in a human patient would be transformative. For trauma surgery, when a patient arrives in an emergency room with a gunshot wound to the heart, or a traumatic brain injury, or a ruptured aortic aneurysm, the race begins. The race is against two things simultaneously: the loss of blood that is occurring right now, and the damage that will occur to oxygen-deprived tissues the moment blood supply is interrupted. To stop the loss, some trauma surgeons already use a procedure called emergency preservation and resuscitation in patients who have suffered cardiac arrest from traumatic blood loss and cannot be saved by conventional resuscitation. The patient is rapidly cooled using ice-cold saline solution. The heart is stopped entirely, and the metabolic demands of the tissues fall dramatically as the temperature drops. The surgical team then has perhaps 1 to 2 hours to repair the damage before the patient is rewarmed and resuscitated. The technique remains experimental, but early results have been promising enough to continue development. True induced hibernation, the ability to reduce a patient's metabolic demands, not just through cooling, but through active biochemical suppression. The way a ground squirrel suppresses its own metabolism would potentially offer longer preservation windows, more stable physiological states during the suppression, and faster, safer recovery. All of these would be enormous advances. Organ transplantation represents another frontier. The limiting factor in transplantation is often time, the window during which a donor organ can be preserved and transported before damage makes it unusable. Current preservation solutions can maintain hearts and lungs for only 4 to 6 hours. kidneys and livers last somewhat longer. A technique that induced metabolic suppression in the donor organ, reducing its energy demands during the preservation period, protecting its cells from the damage of cold storage and oxygen deprivation, could extend these windows significantly. The implications for who can receive a transplant and from where are enormous. Currently, donor hearts and lungs must be transplanted into recipients within a fairly small geographic radius of the donor, extending preservation time by even a few hours would expand the recipient pool substantially, extending it by many hours or days. The kind of preservation that induced torpor might theoretically allow would transform the logistics of transplantation entirely. Space travel is perhaps the most publicly discussed application, and it is worth examining both the appeal and the reality for human space travel beyond the solar system, or even for very long journeys within it, to Mars or the outer planets. The problem of time is severe. Humans cannot be maintained in normal physiological condition for the years or decades that interstellar travel would require, given current propulsion technology, the resources required, food, water, the psychological toll of confinement, scale linearly with the time spent en route. If astronauts could be maintained in a state of metabolic suppression for months or years, consuming minimal resources and experiencing minimal psychological time passage, the calculus of long-duration space travel would change fundamentally. Various space agencies and private organizations have explored this possibility, and while it remains genuinely speculative, for human application, the scientific basis for believing it might eventually be achievable is real. The biology of hibernation provides the existence proof. Metabolic suppression deep enough to sustain a mammal through months of inactivity is achievable. The question is whether equivalent depth can be achieved in non-naturally hibernating species through pharmacological or genetic intervention. The research into aging and cell preservation connects to all of this as well. Many of the cellular stresses that accumulate over a lifetime, oxidative damage, protein misfolding, DNA damage, inflammation, are slowed dramatically in cells that are metabolically suppressed. Some researchers have noted that hibernating animals, which spend significant fractions of their lives in a state of dramatically reduced metabolism, do not appear to age at the accelerated rate that their fast metabolisms during active periods might predict. The suppression of metabolism during hibernation may effectively pause the cellular clock, and understanding the mechanisms by which this is accomplished could have implications for understanding and potentially modifying The rate of aging. This is, to be clear, speculative science. The leap from understanding how hibernators protect their cells to developing therapies that meaningfully slow aging in humans is enormous, and the history of medicine is littered with promising biological observations that did not translate into therapeutic breakthroughs. But the observations are genuine, and the research is active, and the curiosity is fully warranted. What hibernating animals have done is demonstrate to science that metabolic flexibility, the ability to dramatically reduce and then restore the rate of cellular function, is achievable in mammalian physiology. That demonstration in itself is one of the most significant observations that natural history has ever contributed to medicine. Winter is changing. This is not a political statement, or a prediction, or a concern about what might happen in some distant future. It is an observation about what is already measurable, already documented, already showing up in the biology of animals, whose lives are precisely synchronized to the rhythms of the seasons. The average winter temperatures across the northern hemisphere have risen by measurable amounts over the past several decades. The timing of seasonal transitions, the date of the last hard frost in spring, the date of the first snow in autumn, has shifted in many locations by days or even weeks relative to the patterns that prevailed in the mid-20th century. The snow cover that insulates hibernation dens has changed in depth, in duration, in consistency. The availability of food in late summer and early autumn, the food that hibernating animals must consume to build their fat reserves, has changed as the timing of plant productivity shifts in response to warming. All of these changes affect hibernators, and because hibernation is a strategy of extremely precise synchronization between the animal's internal seasonal clock and the external conditions, it has evolved to expect even moderate changes in the external environment can create significant mismatches. Consider the yellow-bellied marmot, a large ground squirrel that lives in alpine meadows of the western United States. Over a period of several decades, researchers have documented a substantial shift in the timing of marmot emergence. From hibernation in spring, marmots are emerging earlier, triggered by the earlier arrival of warm temperatures. But the grasses and other plants they depend on are also growing earlier, and in some cases, the early emergence of marmots is actually tracking the food supply reasonably well, allowing them to capitalize on a longer growing season. This is a relatively optimistic example of ecological flexibility. An animal whose biology has some capacity to respond adaptively to changing conditions. But not every hibernator is this flexible, and not every ecological relationship adjusts so neatly. The timing of insect emergence is critical for bat species that must build their fat reserves in autumn. Before hibernation, bats, with their long hibernation periods and tight energy budgets, are particularly vulnerable to mismatches between their emergence timing and the timing of their insect prey. If insects emerge earlier in spring, but bats do not adjust their emergence accordingly, if the bat's internal clock is less responsive to temperature cues than the insect's development is, then there is a gap. The bat emerges when it has always emerged, and the best of the early insect abundance has already passed. Such phenological mismatches between the timing of different species in an ecological system, are being documented across many ecosystems. As climate patterns shift, the concern among ecologists is not just that individual species might struggle, but that the intricate timing relationships between species in an ecosystem might be disrupted in ways that cascade through entire food webs. For bears, the changing seasons create a different set of pressures. Bears emerging from winter dens increasingly find landscapes that are warmer and snowless earlier than their evolutionary programming expects. In some areas, this has allowed bears to remain active later into autumn and emerge earlier in spring, potentially benefiting those with the behavioral flexibility to take advantage of a longer growing season. But it also brings bears into contact with human settlements more frequently, both because bears are active longer and because the food resources that bears rely on in autumn are sometimes disrupted by changing climate conditions, pushing bears to seek food in unexpected places. The ecological ripples of changing hibernation timing extend to plants as well. Many plant species in temperate and alpine ecosystems depend on large herbivores, including hibernating species like marmots and ground squirrels, for seed dispersal, for disturbance of vegetation that creates germination sites, for the turnover of nutrients that their burrowing creates. When these animals shift their patterns of activity, the plants feel it. What is striking, looking across the research, is how interconnected all of this is. Hibernation is not just a strategy employed by individual animals. It is a thread woven into the fabric of entire ecosystems. Its timing, its duration, the species that employ it, all of these are part of the seasonal rhythms that structure communities of organisms across vast portions of the Earth's surface. The response of ecosystems to climate change is not a simple story of winners and losers. It is a story of complex, nonlinear adjustments in systems with millions of years of evolved relationships, systems that have proven adaptive and resilient across many previous climate transitions, but which are now facing changes, arriving faster than most evolutionary processes can readily track. What we can observe with the careful attention that ecology allows is the system in motion, adjusting, partially adapting, experiencing some losses and some unexpected gains. The hibernators are part of this observation. Their emergence timing, their body condition, their reproductive success, all serving as sensitive indicators of how a changing winter is filtering through the biology of animals that have staked their survival on their ability to wait it out. Let us step back from the laboratory and the field study for a moment. Let us allow the biology to settle and look at what it suggests beyond itself. There is a way of thinking about hibernation that does not require microscopes or metabolic chambers or genetic analysis. It requires only the willingness to observe what a sleeping animal is actually doing, stripped of all technical language, and to ask what it means. An animal that hibernates is an animal that has learned through the long, impersonal education of evolution that it does not always need to be productive. That rest is not the opposite of survival. That stillness, under the right conditions, is the most effective strategy available. That there are times when the wisest thing a body can do is to stop, to conserve, to wait. This is not a small lesson. It runs counter to nearly every value embedded in modern human culture. We live in a civilization that has decided, more or less explicitly, that productivity is the primary measure of value. That rest is something to be earned through prior effort and spent frugally before returning to effort. That growth, economic growth, personal growth, professional advancement, must be continuous and linear, and that any departure from continuous growth is a failure or a loss, or at least a deviation that requires justification. Winter is not a part of this worldview. Winter, in the economic sense, does not exist. There are no seasons. In a global economy, no months of darkness and cold, during which it is appropriate to slow down and wait. The market does not hibernate. The feed does not hibernate. The expectations of others do not hibernate. And yet, the biology of life on Earth suggests that rest, genuine, deep rest, not merely sleep, but strategic withdrawal from the demands of activity, is not a luxury, it is a requirement. The hibernating ground squirrel does not rest because it has earned rest. It rests because not resting would kill it. The rest is the strategy. The stillness is the achievement. Nature moves in cycles. This is among the most fundamental observations anyone can make about the living world. Day and night, the tidal rhythm, the annual cycle of seasons, the longer rhythms of ice ages and warm periods, the cycles of individual organisms, growth, reproduction, senescence, and death. Life does not move in a straight line. It pulses. It oscillates. It returns periodically to states of lower activity before surging forward again. The hibernating animal is the most vivid, most literal embodiment of this cyclic principle. Its body is not merely resting. When it hibernates, it is resetting the physiological processes that accumulate during active life. Oxidative stress, cellular wear, the accumulation of metabolic byproducts are, during hibernation, in some ways reversed or cleared. Some research suggests that the brains of hibernating animals show evidence of being in better cellular condition at the end of the hibernation period than at the beginning, as if the rest has not merely paused the aging processes, but partially reversed some of their effects. There is something in this that speaks to a human experience many people recognize, but find difficult to articulate. The sense that genuine rest, not distractingly active rest, but genuine stillness, provides a kind of restoration that mere reduction in activity does not. The creative person who finds their best ideas come after periods of deliberate stepping back from the work. The athlete who finds that the recovery period is as essential as the training. The grief that must be sat with, not rushed, not productive, not hurried through to something more convenient. These are not hibernation in any biological sense, but they rhyme with it. They suggest that the wisdom built into a squirrel's winter sleep might not be entirely foreign to human experience. We are, after all, mammals. We share the ancient evolutionary heritage of seasonal rhythms, of circadian clocks, of the physiological distinction between day and night, winter and summer. Our melatonin levels still rise as nights lengthen. Our mood and energy still respond, in many people, to the amount of light available. Our biology still carries the traces of a time when winter meant resource scarcity and when the body needed to adjust. Perhaps the impulse, so familiar to so many people in the northern winter, to slow down, to sleep more, to stay indoors, to turn inward. Perhaps this is not a failure of willpower or a symptom of depression, but the echo of something much older, the body remembering, in its dim ancestral way, that winter is the season for a different kind of being, less doing, more stillness, a different pace. Hibernation teaches us, if we are willing to learn from it, that life is not one speed, that survival is not always about moving faster, or working harder, or producing more. That sometimes the most sophisticated response to difficulty is to become as still as possible, to conserve what you have, and to trust that the season will change. The ground squirrel under the snow does not worry about what it is missing. It does not check its status in the social hierarchy of the meadow. It does not feel the pressure of comparisons with other ground squirrels who might be more productive or better prepared. It is simply here, breathing once per minute, maintaining its essential structure, waiting, and in spring, when the light returns and the temperature rises, and its brown fat fires its little furnace back to full capacity, and it climbs out of its burrow into the new world. It is ready, not despite the winter, because of it. This is what rest, at its deepest and most necessary, accomplishes. Not the absence of life, the preparation for it. We are, in the context of understanding hibernation, still early in the story. The phenomenon has been observed for as long as humans have lived, alongside temperate zone animals. The systematic science of it dates back perhaps 200 years. The molecular biology, the investigation of which genes are active during hibernation, which proteins are changed, which biochemical pathways are altered, has been possible for only a few decades, and has accelerated dramatically with the advent of modern genomic and proteomic tools. What we have learned in those decades is remarkable. What we have yet to learn is larger still. The genetics of metabolic suppression is one of the most active and promising areas of current research. Whole genome sequencing of hibernating species, ground squirrels, bats, bears, dormice, hedgehogs, has allowed researchers to identify genes that are uniquely present or uniquely modified in hibernators. compared to their non-hibernating relatives. Some of these genes appear to be genuinely novel, unique to hibernating lineages, with no obvious counterpart in non-hibernating mammals. Others are versions of genes found in all mammals, but with specific mutations or expression patterns that appear to have been shaped by selection for hibernation. Understanding these genetic differences in detail may eventually allow researchers to answer the fundamental question: what is the minimum genetic change required to enable metabolic suppression in a mammal? Is hibernation the product of dozens or hundreds of genetic changes, each essential, or is there a small number of master regulators whose modification could enable the basic capacity in a non-hibernating species? If the latter, if induced metabolic suppression in non-hibernating mammals is achievable through manipulation of a manageable number of genetic targets, then the medical applications discussed earlier become considerably more accessible. Gene therapy approaches or pharmacological agents targeting the key regulatory proteins could potentially induce controlled torpor states in patients or in isolated organs. On demand, the research into neuroprotection that hibernation provides is particularly exciting from a medical standpoint. The brain's ability to dramatically reduce its neural activity during torpor and to restore full function upon arousal without the kind of damage that similar conditions would cause in non-hibernating mammals has obvious relevance to stroke treatment, traumatic brain injury, and the understanding of neurodegenerative diseases. One specific area of interest involves the tau protein, a protein involved in maintaining the structure of neurons that, when it becomes abnormally modified, is associated with the formation of the neurofibrillary tangles, characteristic of Alzheimer's disease and other dementias. During torpor, tau in the brains of hibernating animals undergoes a modification called hyperphosphorylation. The same modification seen in early Alzheimer's disease, but in hibernators, this modification is fully reversed during arousal with no apparent neurological damage. Understanding how hibernating animals reverse tau hyperphosphorylation could provide insights into how to prevent or reverse the pathological process in human brains. Cryobiology, the study of life at very low temperatures, owes an enormous debt to hibernation research. The mechanisms that allow hibernating cells to function at temperatures approaching freezing, and the mechanisms that allow wood frog cells to survive actual freezing, are directly relevant to the challenge of cryopreservation, preserving cells, tissues, and potentially whole organisms at very low temperatures for extended periods. Current cryopreservation technology can preserve sperm, eggs, and embryos reliably, but more complex tissues and organs are much more difficult to preserve without damage. The ice crystal formation that occurs during freezing, the same challenge that wood frogs address through glucose cryoprotection and ice-binding proteins, is the primary obstacle. Learning from the biochemical strategies that cold-hardy organisms use to manage this problem could lead to improved cryopreservation protocols with wide applications in medicine, conservation biology, and potentially long-term storage of biological materials. Aging remains one of the most tantalizing frontiers. The relationship between metabolic rate, oxidative stress, and lifespan in hibernating animals is complex and still not fully understood. Some hibernating species do appear to have longer lifespans relative to their body size than their non-hibernating relatives, which is consistent with the hypothesis that the reduced metabolic rate during hibernation reduces the accumulation of oxidative damage. But the relationship is not simple, and other factors, immune function, reproductive strategy, predation risk, complicate any straightforward correlation between hibernation and extended lifespan. The future of hibernation research is likely to be shaped by two converging forces: the continuing development of molecular tools that allow ever more detailed investigation of what happens at the cellular and genetic level during hibernation, and the increasing urgency of medical and technological problems, trauma care, organ preservation, space travel, aging, that hibernation biology might help address. The animals themselves, the ground squirrels, the bats, the dormice, the wood frogs, are the libraries in which the knowledge is stored. They have been doing this for millions of years, solving problems that we have barely begun to articulate. The task of science is to read what they have written and to understand what it means. Return with me now to the forest. The light is leaving. The pale winter sun that spent the short day barely clearing the tree line has begun its early descent, and the sky above the bare branches is turning through shades of pewter and rose, and then to the deep blue-gray of a winter evening. The snow is still. The temperature is falling. The forest at dusk in winter is a place of extraordinary peace, not the peace of emptiness. We know now that this landscape is full, beneath its surface, with the slow fire of life. But the peace of scale, of stillness, so complete that it enlarges everything it touches. The single crack of a branch, somewhere across the valley, sounds distinct and finite in the cold air. A light wind moves through the uppermost branches, and the snow they carry falls in a soft, dispersing shower, catching the last light as it descends. Beneath the snow, beneath the frozen leaves, and the packed earth, the heartbeats continue. One beat, a pause, another beat. In the burrow of the 13-lined ground squirrel, the body is arranged in a perfect curl, nose tucked beneath the tail, the fur dense and warm between itself and the cold. Its temperature is 4 degrees Celsius. Its heart has beaten 3 times in the last minute. Its breath, when it comes, which is perhaps once every 3 minutes, makes no visible mark in the cold air of the burrow. Because the burrow is cold too, and the distance from the animal's core to its surroundings is small. The fat beneath its skin is slow, molecular work. Triglycerides, broken down into glycerol and fatty acids, carried in the blood to mitochondria in every cell, converted there to the ATP that keeps the membrane potentials maintained, the cellular machinery from ceasing, the minimum fires from going out entirely. The fat is warmth. The fat is time. The fat is spring. In the cave on the hillside, the bats are similarly suspended, their leathery wings drawn close, their echolocation silent, their navigational brains idling at a fraction of their summer capacity. They dream of nothing. They plan nothing. They simply are, in a state that is less than life, as we usually conceive it, but more than the absence of life. A middle state, a sustained possibility. The bear in her den has shifted in the last hour, turning slowly, rearranging herself, and the 2 cubs pressed against her belly. The cubs are very small still, their eyes not yet open, their thermoregulation not yet reliable. They depend on her warmth, on the heat her body generates, even in its reduced state, on the rich milk she produces, even in the near absence of food. She breathes slowly and steadily. She is present, in some attenuated way, in the den. She knows where she is. She is not gone. She is simply waiting, somewhere across the continent, in a meadow forest in Vermont. A wood frog lies frozen beneath a log. Its heart is stopped. Its blood has become an ice-thickened slush in vessels that carry nothing. The glucose that protects its cells is at concentrations that would read on any blood glucose monitor designed for mammals as a catastrophic crisis. And yet, the cells are whole. The proteins are intact. The DNA is undamaged. The frog is poised on the threshold between the living and the not living. And the threshold is so narrow that biology itself cannot quite determine which side it stands on. Night has fully arrived now. The snow has resumed falling, softly, from a sky with no visible source. The flakes are small and light, the kind that drift rather than fall, suspended for a moment in the cold air, before settling onto the surface of the old snow with no sound at all. The forest fills with this quiet precipitation. The branches collect thin new layers of white. The den entrances, the small holes in the soil, and under the roots, and at the base of fallen logs, are slowly covered, slowly hidden, slowly erased from the surface of the world. This is the genius of hibernation. Not just that it allows animals to survive winter, but that it allows them to survive winter so quietly, so efficiently, so entirely below the threshold of detection, that winter itself does not know they are there. They make themselves into part of the silence. They become indistinguishable from the patience of the landscape. There is no drama in this. No struggle. No heroism in the conventional sense. The squirrel does not fight winter. It does not endure winter in the way a marathon runner endures the last miles of a race. Through sheer will and the suppression of pain, it does something far more sophisticated than willful endurance. It rearranges itself at the molecular level, so that winter presents almost no challenge at all. The cold that would kill a cat or a rabbit or an unprotected human in hours is simply irrelevant to a body running on 4 beats per minute and 1 breath every 3 minutes. The cold passes over the burrow like weather over a stone. The stone does not struggle. It simply persists. And when we understand this, really sit with it, really let the biology settle into its fuller meaning, we realize that what we are observing in hibernation is not an animal making the best of a bad situation. We are observing an animal for whom winter is not a bad situation at all, for whom winter is simply one of the 2 seasons of its life. The warm season of eating and breeding and moving through a sunlit world, and the cold season of the deep, slow, conserving dark. Both seasons are equally part of the animal's life. Neither is a deviation from normality. The hibernation is as natural as the summer foraging. The stillness is as complete an expression of the animal's biology as the running. And then spring arrives. Always, eventually, spring arrives. The days lengthen. The angle of the sun steepens. The soil begins to warm from the surface downward, slowly, degree by degree. The ground squirrel's brown fat receives the signal first. A slight warming of its den. A change in the length of the darkness. Some subtle shift in a signal that its sleeping body has been monitoring all along. The brown fat activates. The furnace begins. The shivering starts. A deep muscular trembling that generates heat and uses it to generate more heat. A cascade of warming that carries the body temperature upward from 4 degrees to 10 to 20 to 35 over the course of a few hours, powered entirely by the fat that has been waiting for this moment since October. The heart quickens. 2 beats per minute, then 5, then 20, then 100, then 200. The electromechanical rhythm of a healthy heart reasserting itself as the temperature climbs, and the molecular machinery of cardiac function returns to its normal operating range. The eyes open. The animal is stiff. It is thin. It is, in many respects, a reduced version of the animal that descended into this burrow 5 or 6 months ago. But it is alive. It is functional. And within a day or two, it will be foraging. Within a week, it will have restored much of its normal behavioral repertoire. Within a month, it will be breeding. And the cycle will begin again. What hibernation is, at its most essential. What it has always been, since the first small mammal of the Eocene shivered and stilled in a cold burrow and waited for warmth, is endurance. Not escape. Not absence. Not the denial of winter. But its acceptance. On terms that life has negotiated very carefully over millions of years. Endurance. The winter is real. The cold is real. The scarcity is real. These things do not become less real because the animal chooses not to fight them directly. The ground squirrel does not pretend that winter is not happening. It simply waits. It makes itself small. It conserves what it has. It trusts the mathematics of fat and time, the reliability of the light returning, the same light that has reliably returned for every winter since the first winter this lineage endured. There is something in this that exceeds the simply biological, that carries into the domain of the quietly philosophical. In a world that measures worth in output, that values constant productivity, that equates stillness with failure. Here is a reminder, written in fur and fat and 4 beats per minute, that sometimes the deepest achievement is simply to persist, to hold on, to reduce oneself to the most essential elements, and wait for the season to change. The snow is still falling. The den is warm enough. The fat will last. Beneath the snow, the hearts beat slowly. Cells conserve their energy. Brown fat waits to become warmth. The biological clock ticks on in the darkness, counting the days, measuring the light, preparing already, even in the deepest winter, for spring. Life does not disappear in winter. It goes quiet. It goes deep. It goes into the earth, into the rock, into the frozen body of a frog beneath a log. And it waits, with a patience that no conscious being could sustain. And when the world turns warm again, as it always has, as it always will, it is ready. The snow above the den settles. The night deepens. The forest is still. And far below the surface, in the warm darkness of the earth, something breathes. Once more. Still here,